For decades, traditional gene therapy has operated on a straightforward premise. If a patient has a broken gene, you add a healthy one to do the job. But dominant genetic mutations require a different approach. In these cases, adding a healthy gene doesn't fix the problem because the original mutated gene is actively causing harm. In diseases like Huntington's or oculopharyngeal muscular dystrophy, the mutation acts like a rogue factory. It aggressively prints misfolded, toxic proteins that accumulate in the patient's cells and slowly destroy surrounding tissue. To stop the disease, you have to shut down the factory. That requires a biological strategy known as RNA interference, or RNAi. Instead of trying to fix the gene itself, RNAi works as an interception system. It hunts down the messenger RNA, the genetic courier carrying the instructions to print the toxic protein, and destroys it before it ever reaches the ribosome. The most common way to trigger this interception is by injecting synthetic small interfering RNAs, or siRNAs. They do the job well, but they have a critical biological flaw. They degrade rapidly in the bloodstream and within the cells. Because these are lifelong genetic diseases, keeping the toxic proteins at bay with synthetic siRNA requires constant, unending readministration. The medicine simply fades away. To solve that fading problem, genetic engineers pivoted to a structural alternative, short hairpin RNA, or shRNA. Instead of repeatedly delivering temporary doses of synthetic RNAi medicine, the shRNA approach delivers a permanent genetic blueprint. Once installed in the nucleus, this blueprint continuously generates perfectly folded, hairpin-shaped RNA structures. By integrating the instructions directly into the target tissue, shRNA turns the patient's own cells into self-sustaining medicine factories. It solves the longevity problem with a single treatment. Getting that blueprint inside the cell is the first major hurdle. Naked shRNA plasmids degrade almost instantly in the human bloodstream. To survive the journey, they are loaded into viral vectors, like adeno-associated viruses, which act as microscopic, highly targeted delivery trucks. The viral vector navigates the bloodstream, latches onto the target cell's membrane, and injects the shRNA blueprint directly into the nucleus. Inside the nucleus, transcription begins. The cell's own RNA polymerase reads the newly delivered blueprint and extrudes a single strand of RNA. Because of strict sequence constraints, this strand folds back onto itself, locking into a distinct hairpin shape. Next, exportin 5 grabs this hairpin, shuttles it through a nuclear pore, and releases it into the cytoplasm. At this point, the cell has successfully manufactured and deployed the raw shRNA weapon, but in its hairpin form, it is completely unarmed. To arm the shRNA, the cell relies on an endogenous enzyme called dicer1, which functions essentially as a pair of molecular scissors. Dicer1 recognizes the hairpin, measures it, and chops the loop off, creating a short double-stranded RNA molecule. This is fed into the RNA-induced silencing complex, or RISC. RISC splits the strands, discards the weaker one, and locks the other in place as a guide. Driven by a catalytic engine called argonaute-2, or AGO2, the fully armed RISC complex now begins its hunt. It uses the exposed sequence on the guide strand to rapidly scan the cytoplasm, searching for the matching disease-causing mRNA. When it finds a perfect sequence match, AGO2 locks onto the target mRNA and cleaves it directly in half. The genetic instructions are instantly destroyed. This hijacking of the cell's natural processing machinery prevents the toxic protein from ever being printed. Moving this mechanism from a lab setting to a human patient introduces a massive clinical hurdle. Most patients suffering from dominant genetic diseases possess one mutated allele creating toxic proteins and one healthy allele producing proteins they need to survive. If you deliver a standard shRNA, it will indiscriminately wipe out all mRNA associated with that gene. Destroying both versions simultaneously eliminates the disease, but leaves the patient entirely deprived of a vital biological function. To bypass this friendly fire problem, engineers testing treatments for Huntington's disease developed a strategy called allele-specific silencing. The solution relies on targeting a single nucleotide polymorphism, or SNP. An SNP is a tiny one-letter genetic typo present only on the mutated allele. By designing the shRNA guide strand to perfectly match that exact typo, it physically rejects the healthy mRNA. Targeting a single mutated letter turns a broad-spectrum genetic weapon into a highly precise sniper rifle. It selectively eliminates the disease-causing mRNA while leaving the healthy biological function intact. However, in conditions like oculopharyngeal muscular dystrophy, a viable SNP isn't always available to target. When sequence-level discrimination is impossible, geneticists use a dual-action platform known as silence and replace. First, the viral vector delivers a hyperpotent, non-discriminating shRNA that wipes out all endogenous mRNA for that specific gene. The mutant and the healthy transcripts are both eradicated. But inside that exact same vector is a second payload, a newly engineered, perfectly healthy replacement gene. This codon-optimized transgene has been structurally altered so that it is immune to the co-expressed shRNA. As the native mRNA is destroyed, the shielded replacement immediately repopulates the space with healthy instructions. Mastering the delivery and execution of shRNA provides a permanent mute button for humanity's most devastating genetic errors.